Hello friends. Today we are going to get idea about very interesting practical number 8 physiology demonstration experiments. Friends, we have to keep in mind today we have to get idea about physiological experiments in the sense how the things are going to happen in the nature that we can prove experimentally here. Now, for that one, first we have to get idea about imbibition or imbibition in seeds. Second, transpiration pool experiment, arc oxynometer, which is used to measure the growth. Osmosis, that is curling experiment. Effect of auxin on root formation. Then instrument also we have to study, which are very much useful in physiology of a plant or to study the physiology of the plant. In that spectrophotometer, portable leaf area meter, conductivity meter, and centrifuge. My friends, what is the principle? In case of the physiological experiment, we should first of all understand very well the principle. So, principle of imbibition in seed, first our demo experiment is that absorption of a solvent by colloidal material resulting into swelling is called as a imbibition. Once again, try to understand absorption of a solvent, isn't it? Absorption of a solvent by colloidal material resulting into swelling is called as a imbibition. General example I will give to you. We know that actually wood shows, wood in the sense door, which is, we have to say that, prepared with the help of a door, with the help of a wood. If that door gets, we have to say that, we can find the swelling in that door during the rainy season if water gets comes in contact with that door itself. Is that so? That is because of some colloidal material present and solvent, that is the water, which is absorbed by that colloidal material and results into swelling of that door, isn't it? So in rainy season, most of times when we close the door or open the door, at that time that becomes too much tight. That tightness is due to the swelling, and swelling is due to absorption of a solvent, that is water, by the colloidal material present in the wood itself, isn't it? So this is the best example. So imbibition is nothing but absorption of a solvent by colloidal material resulting into a swelling is called as imbibition. Now, which is that colloidal material? So we know that in case of proteinaceous seeds have a very high imbibing capacity. Means in the seed actually protein or reserve food material is a protein is there, and that proteinaceous seeds have a very high imbibing capacity. Much pressure is exerted due to the imbibition, which is called as imbibition pressure. My friends, we know that because of swelling, some pressure get exerted, and that pressure is also known as a imbibition pressure. The pressure which is created because of absorption of a solvent by colloidal material. Here, requirement for this physiological experiment, demo experiments are: first, two test tubes having same size, same thickness of that one, same, we have to say that, length, each and everything, with cotton plug. Cotton plug we have to use here, where plastic caps we are not going to use, we have to use the cotton plug. Dry seeds of gram or pea and water, these are the only requirements of this experiment. What we have to do, what is the procedure of this? So first of all, fill up one test tube with the dry seeds. You have to take out of two test tubes one test tube, and that is fill up with the only dry seeds. Fix the cotton plug to the mouth and then close that seed filled test tube mouth with cotton plug. Okay, now here, keep both the tubes, second test tube is there in our hand, and that test tube, first of all, fill with dry seeds and add a sufficient water. Add a sufficient water and fix the cotton plug perimeter. Why we have to use the cotton plug? We know that when we will add water into that one, seeds respires itself, respiration process. And the actually colloidal material, proteinaceous material present in the seed absorbs the added water in second test tube. The test tube we are going to fill with the first of all dry seeds, and then we are adding sufficient amount of water into that one, and then the mouth is closed with the cotton plug. So here what happens actually, keep this both the test tube overnight. We have to keep that test tube as it is for 12 hours, or we have to say that 18 hours. Then what happens, which observations we have to note? No change in the first test tube. And diagram also, sketches also, will be able to see the things. The first test tube, that is first test tube, have no any type of a cracks on that test tube, but here second test tube is cracked, broken. Second test tube cracked or broken due to some pressure is exerted by the imbibed seeds. In the sense, seeds plus water, that solvent absorbed by colloidal seeds, and seeds started swelling, and that swelling created the pressure. That pressure is known as a imbibition, or I will say that the solvent absorbs the solvent absorbed by the colloidal material, resulting into swelling, and that swelling leads, that process is known as imbibition, isn't it? The seeds absorbs the solvent, results into swelling, is called as imbibition, and the pressure created due to imbibition is called as a imbibition pressure, and that pressure results or created is some force, and because of that force the test tube, glass test tube, get cracked on. Here most important thing is that don't use the plastic test tube, we have to use the glass test tube, that is most important. So what is the conclusion out of this? So conclusion out of this is that proteinaceous seeds imbibe water and swells. This absorption of water by the seeds actually creates imbibition pressure, and it results in cracking of a second test tube, isn't it? After getting idea about first demonstration physiological experiment, that is of imbibition in the seeds, we can get idea about second, that is a transpiration pool experiment. My friends, we know that what is the transpiration, that is nothing but loss of water in the form of water vapors through the aerial parts of plant body is known as a transpiration. Now that transpiration process creates some pull, and that we have to say, is it possible or not? That also we can see with the help of this demo experiment. What is the principle of that? Water is a continuously evaporated from the aerial parts of the plant body during the process of transfer. Continuous process, that is a loss of water in the form of water vapors through the aerial parts of plant body. This loss of water by aerial parts is compensated by the water absorbed through the roots due to suction force or pulling force. My friends, you will have to keep in mind this is a continuous process, isn't it, of a absorption of a loss of a water. Now whenever loss of water takes place by the aerial parts of plant body, immediately that is compensated by the water absorbed through the root due to the suction force or pulling force. कंटिन्युअस जस्ट जमिनीमध्ये पाणी आहे ते जमिनीतलं पाणी परत रूटमध्ये घेतलं जातं. इट फॉर्म्स वी हॅव टू से दॅट द कंटिन्युअस कॉलम्स इन टू द झायलॅम, isn't it? So suction is thus a sucking activity. It is because of formation of a partial vacuum in the inner space, so that the external atmosphere pressure forces water into the vacant spaces, isn't it? My friends, you'll have to keep in mind how the things are going to happen. So whenever loss of water takes place through the aerial parts of plant body in the transpiration physiological process, At the same time, suction force is created, pulling force is created due to this transpiration process, and atmospheric pressure responsible just to fill up the vacant places. And that is the actually, we have to say that, my friends, principle of this transpiration pull experiment. Which things are required? Requirement is potometer, capillary, rubber tube, stand, beaker, mercury, and corks. These are the requirements. Now, what is the procedure? Take a potometer, fix it to the stand. Isn't take a potometer, fix it to the stand. At the lower end of the potometer, a rubber tube is fitted, having a capillary to its other end. Having a capillary to its other end. Fill the photometer carefully with water. Fill the photometer carefully with water by holding the end of capillary by thumb. By holding the end of capillary by a thumb. Fit the cork tightly in the upper end of photometer and insert the plant twig into the cork. Into the cork. Take some mercury in the beaker. Now in this mercury, keep the lower end of the capillary tube. Mark the level of mercury in capillary tube. Make the apparatus airtight. Keep it in the sunlight and observe after some time. My friends, very simple procedure is this one, but one by one you will have to understand what we are going to do, what we have to do, and why to do, isn't it? In case of science, when you will ask the questions like what, when, why, if not done, what may can happen, if done, what can takes place. If you raise such type of questions, immediately you will get the solutions from that questions, and definitely the concept may can get clear. So this is the actually, in case of science, science students should have inquisitive mind. They will have to always ask the questions. So this is the technique of learning of a science itself. In this transpiration pull experiment, what the things that we have to do, in our hand we are having a potometer that is to be fixed to the stand. At the lower end of that potometer, we have to attach, we have to fit the rubber tube, but that rubber tube is having a capillary tube to its other end, isn't it? Now, fill the potometer carefully with the water by holding the end of capillary by a thumb. Fit the cork tightly in upper end of a potometer and insert the plant twig into the cork. Take some mercury in the beaker. Now in this mercury, keep the lower end of the capillary tube. Mark the level of mercury in a capillary tube. Make the apparatus airtight. Keep it in the sunlight and observe after some time, my friends. Here you will be able to see in the sketch each and everything, isn't it? Each and everything. One by one, I will give you idea how to set up this physiological experiment just to understand the suction due to transpiration process. Here, as we have seen, actually which things are required to us. First of all, we should have in our hand beaker. Then we should have mercury. Then we should have rubber tube. Here at the one of the end, lower end of the rubber tube, there is a capillary. Then this instrument totally with, so this is actually potometer. This is a potometer in which water we have added. Cork is there and healthy plant twig is inserted into that cork, my friends. And now the things are, means actually potometer is we have fitted to this stand. So this is the requirement. What we have to do, first of all, fix the potometer to the stand. Yes, we have fixed that one, isn't it? And this potometer actually having which things, rubber tube at one end, and another end of this rubber tube we have attached a capillary, isn't it? Now what we have to do, first of all, we have to first of all fill up this potometer with the water, but lower end we have to fix with the thumb, isn't it? Now after filling the water, what we have to do, immediately we have to dip the lower end into the mercury. Simultaneously, this twig, not only in the last lecture, in the last not lecture, in the practical, I have to say that study of a transpiration, rate of transpiration that we have studied. In that one, how we have cut the twig, healthy twigs, we have to cut under the water, slanting cut, and immediately we have to insert into the this potometer through the cork. Now this is the set of now after setting this experiment we have to keep this experiment in a sunlight. So what happens is because of sunlight temperature may increase, and due to that one process of transpiration starts, or we have to say the rate of transpiration may can increase, and once the rate of transpiration increases, continuous loss of water will takes place through the stomata, or we have to say that cuticle, and that water loss immediately will be compensated by the water present in this potometer. Now as the water will move upward, definitely here the capillary, in the lower capillary there is a rise in the mercury level takes place, isn't it? Rise in the mercury level in capillary, that indicates that the vacuum, I have to say that the some space is developed, and that empty space is filled by the mercury, isn't so? Continuous loss of water, that water this twig will absorb from this potometer, and when there is a some space is there, vacant space is there, that can be compensated or filled by the mercury, so that will give us idea that is some pool, transpiration pool is created, and that transpiration pool is responsible for absorption of a water, isn't it? So suction pressure is created, and that suction pressure is responsible for that. So what is the observation? There is a rise in the mercury level in the capillary tube. There is a rise in the mercury level in a capillary tube when we will set up this experiment in the sunlight, expose this experiment to the sunlight. Now what are the conclusions? The rise in mercury level is because of uplifting of a water column in the xylem of aerial organs by the transpiration process. So water is lifted in upward direction and space is created. Water is lifted in upward direction and space is created in the capillary tube, which is occupied by the mercury in the beaker. So this is we have to say very interesting experiment is there, my friends. Keep in mind, you will have to, you can do that experiments at our places, although with the things which are available to us at our places. Now third experiment, that is a arc indicator. That is a arc indicator. The arc indicator is used to measure the growth of plant, isn't it? Growth of plant. Now what is the growth? Growth is nothing but the permanent and irreversible increase in size. Permanent and irreversible increase in size, isn't it? That is known as a growth. That is known as a growth itself. The growth is a greatest at the stem tip. The growth actually takes place at the tip of the cell, at the tip of the plant, shoot of the plant. So there are, we know that meristems are present, and meristem tissue having capacity to undergo division and redivision, isn't it? And because of that one we will be able to see that the growth rate is a very fast at the stem tip. Keep in mind, ग्रोथ इज अ परमनंट आणि इरिव्हर्सिबल. का म्हटलं परमनंट आणि इरिव्हर्सिबल? आपण असं कधी बघितलं का एखादं झाड 1 वर्षापूर्वी 7 मीटर होतं आणि पुन्हा 3 मीटरवरती आलं? एकदा ते 7 पासून पुढं गेलं, 7.5 मीटर गेलं तर 7.5 मीटर राहील एखाद्या वेळेस, तिथून पुढं जाणार नाही, पण त्याची कमी होणार नाही उंची. सो दॅट इज द ग्रोथ. हाईट असेल, गर्थ ऑफ प्लांट असेल, आपण कधी बघत नाही, आणि इरिव्हर्सिबल आहे, ते पुन्हा खाली येत नाही. सो इन्क्रीज इन द साईज. कंटिन्युअस चेंज इन द हाईट कॅन बी मेजर्ड बाय आर्क ऑक्झॅनोमीटर, आर्क इंडिकेटर. आर्क इंडिकेटर हे इन्स्ट्रुमेंट आहे. ह्या इन्स्ट्रुमेंटच्या सहाय्यानं विल बी एबल टू मेजर द ग्रोथ, height, change in continuous of height of the plant. A potted plant, arc indicator, a weight, a string stand, isn't it? Now, after getting idea about the requirements, the procedure, first of all, we have to tie a thread to the tip of potted plant. Why to the tip of potted plant? Because we know that meristems are present that undergo division and redivision. Below shoot tip, we will attach that one thread. Now, friends, keep in mind here in the requirement we have written the string, isn't it? And here we are writing tie a thread and string same one. Okay, the tie a thread to the tip of the potted plant, fix it to the pulley of the arc indicator by weight to which a long needle or pointer of the indicator is attached. Record the initial reading on a scale where the pointer is steady. Allow the plant to grow for a few days. When there is an increase in the height of the plant, the pointer moves over the graduated arc indicator. Record the movement of pointer at regular intervals. What observations we have to make? See, that is because of growth of plant, the pulley rotates in a downward direction. Because of growth of plants, the pulley rotates in downward direction. It causes the movement of pointer on the arc scale. It causes the movement of pointer on a arc scale. This movement is proportional to the growth, my friends. Proportional to the growth rate of the plant. This movement is proportional to the growth rate of the plant's. Conclusion: by recording the movement of the pointer by angular degree at particular intervals. By recording the movement of the pointer by angular degrees at particular intervals, the growth rate of the plant can be measured. So this is the methodology. Now, this picture gives you idea how that arc indicator is there and how it works is there. My friends, in case of this arc indicator, no doubt this is the total arc indicator is there, isn't it? Now, here potted plant we have taken, potted plant is there, and we have tied thread or string to the shoot tip of a potted plant, and that string we have passed on pulley. This structure is a pulley. So at the one end of the thread there is a shoot tip where we have tied that on, another end there is a weight is there. So my friend, as the growth takes place, this string actually moves downward, means this pulley actually moves downward, and in proportion of the growth, in proportion of a growth, we have to say that this pointer also moves, isn't it? This pointer moves, and this graduated, we have to say that scale is there, and that scale we will be able to see, we will find out the growth rate of a plant, isn't it? So this is the very simple experiment. Although it is a time consuming, we can get idea about how to measure the growth rate of the plant with the help of a arc indicator. If you have any difficulty about this, you can ask me. Now friends, we have to get idea about fourth physiology experiment, demonstration experiment, that is osmosis. Now what is osmosis? As in the last theory lectures we have got idea about, the osmosis is a special type of a diffusion. Special type of a diffusion. But the diffusion in this special type of a solvent molecule moves across the semi-permeable membrane from its region of a lower concentration to region of a higher concentration. Here you will have to keep in mind the diffusion of a solvent molecule across the semi-permeable membrane from its region of a lower concentration to the region of a higher concentration is called as osmosis. Am I right? Friends, I have tell you one important thing is that water moves towards the sugar. पाणी कुठं जाते साखरेकडे जाते. That only formula you have to keep in mind in case of osmosis. So there is a movement of a solvent from region of a lower concentration to the higher concentration, isn't it? But that solvent molecule moves across the semi-permeable membrane from its region of a lower concentration to higher concentration is called as osmosis. The plant cells kept in the solution of a lower concentration shows endosmosis. Means water enter inside. So that is endosmosis. That happens only when we place a cell in a solution of a lower concentration. In the sense, the solution in the plant cell is having a higher concentration. Solution in the plant cell is having high as compared to the solution in which that plant cell has been kept. And because of that one, endosmosis takes place, means the water moves from outer side to inner side of the cells, means from outer atmosphere to inner side of the cell. That is known as the endosmosis when kept in the solution of higher concentration. But when we will keep that same plant cell into a higher concentration of its, then what happens? Exosmosis takes place, means water comes out. Water comes out. Requirement: Petri plate, sharp razor blade, water, 0.4 molar sugar solution, 0.4 molar NaCl solution, plant material, fresh petiole of arum or nymphaea. फ्रेश petiole of arum or nymphaea. arum म्हणजे आपल्याला आळू, लक्षात येतं ना, आळूचे आपण पिटिओल घेतलेले आहेत, डेट घेतलेले आहेत. प्रोसिजर: टेक इक्वल क्वांटिटी, that is 10 ml of a water, 0.4 molar sugar solution and 0.4 molar NaCl solution in the 3 Petri plates and label them. Prepare the equal sized 3 pieces of petiole, cut radially longitudinal slits of about 1 centimeter in each piece from both ends. Place the pieces in each solution for 25 minutes and observe the changes in the split end portion. What things have been done that you have to keep in mind. Before that one, some observations we will record here. Observations: in water, the split parts of petiole curls outward. In case of the petiole, in case of water, when we will keep that petiole of a arum, the split part of petiole curl outward, showing the curling due to formation of a tissue tension. This is because the inner cells are more turgid than outer cell. Inner cells absorb more water and become a turgid due to endosmosis. In 0.4 molar sugar solution, the split parts of the petiole pieces in the sugar solution shows no change because inner cell shows exosmosis. In 0.4 molar NaCl solution, the split parts of the petiole piece in this solution shows no curling because exosmosis takes place. Conclusion: curling takes place due to endosmosis. My friends, you have to keep in mind how the things are going to take place in the process of a osmosis. Here, this is the actually sketch which is sketched by the student, and we have taken the photographs for your understanding. As already we know that what we have to do, we have to endosmosis and exosmosis. Here, my friends, curling experiment is that what happens. First of all, we have taken here, this is a piece of a arum, this piece of arum, this piece of arum. This piece of arum, so we have placed into a different concentration solutions. Here, first piece we have kept into a Petri plate, that is of a water. Second, that is a 0.4 sugar, 0.4. Third one in a 0.4 NaCl is there, isn't it? Here, in case of this one, what happens after some time, that Here, curling occurs when we place the arum pieces which are cut having a slits of 1 centimeter. So what happens actually? This curling is due to endosmosis. Curling we will be able to see at the tip. This curling is due to endosmosis. In the sense, the water present into the petri plate is enter the cells present in arum petiole, and because of that, endosmosis takes place. Here, no change is there when we place arum pieces having a slits arum pieces having a slits placed into a zero point 0.4 molar sugar solution and zero point 0.4 molar salt solution, NaCl solution. There is no change in the sense the water present into the arum that has been comes outside, that is exosmosis takes place. Because of that, no curling. So this curling experiment shows presence of a curling is due to endosmosis and no curling is due to exosmosis. So this we have observed here. My friends, this experiment will be kept into the examination to you for the spotting or identifications. When that experiment is there, you have to mention principle of that experiment, requirement, each and everything, and what things are going to observe by you, isn't it? That you have to mention, that you have to study, that you have to write in the examination. Now, effect of auxins on routing. My friends, fifth experiment today we have to learn, that is of a effect of a auxins on routing. We know that establishment of each and every plant depends upon its routing. If routing never takes place, establishment never takes. If routing is too much slow, then also there is establishment is also slow. In that one, some growth hormones are there. And that growth hormones induces or accelerates the root formation. So, which are those? Which type of effect experiment that we have to study under this our demo experiment? Principle: the auxins like the indole acetic acid (IAA), indole butyric acid (IBA), and acetic acid naphthalene acetic NAA are the major phytohormones essential for the growth and development. Auxins are synthesized in a growing apical region, then transferred to the region of their requirement. That is most important. Location, site of the synthesis of growth hormones or auxins is there at growing apical region. Synthesis takes place and that are transported in the plant body wherever that are required. Now, those plants which are propagated by cutting can be treated with the auxin, which has the property to induce the root development when applied in a low concentration. When we apply in a low concentration at that time, that induces the rooting to the plant. So that is most important. We have to say that methodology, or we have to say that property of a growth hormone that is useful on industrial point of view to the fellows, those who wants to multiply the vegetatively growing plant very fast or establish them, or nursery where the plants are produced. My friends, requirement 1000 ppm. ppm means parts per million. ppm च्या फॉर्म्युल्याच्या बाबतीत म्हणजे साधा डिफरन्स लक्षात ठेवा. 0.5%, व्हेरी सिम्पल थिंग इज दॅट. 0.5% इज नथिंग बट द 5000 ppm. 5000 ppm is there, isn't it? Now, here we have to prepare 1000 ppm or 0.1% auxin lanolin paste can be prepared by dissolving 100 milligram of a auxin in a 1 to 2 ml of ethanol, and the solution is thoroughly mixed with the 100 gram of a lanolin, isn't it? So these things are there. Why do we have to dissolve into a small amount of ethanol, then make the volume final volume with the help of water? Because we see 1 to 2 ml in the sense it get dissolved, it never get dissolved into the water. That's first of all we have to dissolve. Lanolin, that is actually a sticky substance, that also we have to add into that one. Cutting or girdle plants of sunflower, tomato, tobacco, or any other suitable plant from the ornamental. Croton is there in our garden, we know that that also grows vegetatively, sorry, multiplies vegetatively. Procedure. A small quantity of lanolin pest 0.1% oxin is applied at the base of the stem cutting or stem girdle. After few days, the observations are made. In case of the plant where the oxin is employed, initiates or even rootlets comes out. Conclusion: oxin has a property to stimulate the initiation of a cell division. Auxin led to the root formation at low concentration. Now, how the root initiation, root development takes place? Actually, let us the formation of a new cells takes place and that division. So here auxin plays important role in case of accelerating the rate of cell division, and that leads to the root formation, isn't it, at low concentration. That is proved. So this is the effect of, we have to say that, auxin on a rooting. Now we have to six, that is a spectrophotometer instrument which is very much useful in plant physiology. Actually, that we have taken into physiology demo experiment also. Here the spectrophotometer. A spectrophotometer is an instrument that measures the amount of light absorbed by solutes when it passes through the sample. So simple thing is there. काय केलं असतं की इथं टेस्ट ट्यूब आहे, त्या टेस्ट ट्यूबवरती ठराविक इंटेन्सिटीची लाईट पाडली जाते, त्याच्यातून पास केली जाते. जसजसं सोल्यूटचं प्रमाण त्या सोल्युशनमध्ये वाढत जाईल तसतसं त्याच्यामध्ये ॲब्सॉर्प्शन, म्हणजे त्याच्यावरती इन्सिडेंट लाईट आहे त्याचं ॲब्सॉर्प्शन वाढत जाईल आणि त्यातनं कमी लाईट पास होईल. सो दॅट कुड बी द ट्रान्समिटेड लाईट. सो दॅट कुड बी द ट्रान्समिशन. आणि मग त्या ह्याच्यावरती त्याचं मेजर केलं जातं. फॉर एक्झाम्पल समजा 30% ॲब्सॉर्ब केली अँड 70% जर ट्रान्समिट केली तर डेफिनेटली विल से दॅट 30% सोल्यूट्स आर प्रेझेंट इन टू दॅट सॉल्वंट, isn't it? So this is the actually simple principle is used here. Every chemical compound absorbs the transmitted light. Very interesting thing is that you have to keep in mind. Every chemical compound absorbs, transmits, or reflects the light in a certain range of a wavelength. Every chemical compound having its property, that is absorbs, transmit, or reflect the light in a certain range of wavelength, isn't it? The principle of this instrument is based on Beer's and Lambert's law. Beer's and Lambert's law, which states that amount of light absorbed by the substance is directly proportional to the concentration of the substance and path length of light passing through the solution. Amount of light absorbed by the substances is directly proportional to the concentration of a substance. म्हणजे मी मगाशी सांगितलं की त्याच्यात किती सोल्यूट्स आहेत त्याच्यावरती त्याचा रेट ट्रान्समिट किती करणार आणि हे किती करणार ते राहतं. On the basis of this law, spectrometer works and measures the concentration of the solute present in the solutions. आता आपल्याला एखाद्या सोल्युशनमध्ये प्रोटीन किती आहे हे काढायचं असेल, एखाद्या सोल्युशनमध्ये शुगर किती आहे हे काढायचं असेल, तर मग अशा वेळेला दिस एक्सपेरिमेंट हेल्प्स टू अस, किंवा अमायनो ॲसिड किती आहे ते मोजायचं असेल आपल्याला. सो दॅट इज व्हेरी पॉसिबल विथ द हेल्प ऑफ द स्पेक्ट्रोफोटोमीटर. We measures the concentration of a solutes present in the solution. Depending on the range of wavelength of light source, it can be classified into two different types. That is visible spectrophotometer and UV. UV spectrophotometer is a little bit advanced level. Now here we will be able to see that two instruments are there. That is a visible spectrometer on upper side and from lower side, that is a UV. So these instruments are used to measure, we have to say that, concentration of a solution. So with the help of that we will be able to find out the amount of solute present in the solvent. Now portable leaf area meter. This also I have to say that most important instrument is there. We have to measure, because leaf area, we know that on the basis of leaf area, or the leaf area is directly proportional to the rate of photosynthesis. So this instrument gives us idea about portable leaf area meter. Size of the leaf we will be able to find out with the help of this instrument. Here, my friends, leaf area, a device is used to measure the leaf area, length, width and perimeter of the leaf. Portable leaf area meter can be used to provide rapid, non-destructive measurement of leaf surface area, length and width and perimeter. Some can also measure the diseased area by altering the sensitivity of a detecting system. This instrument have the ability to record the measured parameters as well as store a digital image of the leaf scanned. Specific leaf area can be used to estimate the reproductive strategy of a particular plant based on the light and moisture level. 40% among other factors. Specific leaf area is one of the most widely accepted key leaf characteristic used during the study of a leaf characters or traits. It is easy to handle and carry during the long distance field work. Now, after this one, conductivity meter. My friends, this is also very interesting instrument. That is, it measures the ability of the solutions to conduct the electricity. In the sense, we have to measure EC, electrical conductivity meter is there. We can measure the salinity status, or we have to say that suppose we are having a solution in our hand, we'll be able to see the salinity of that one in EC. That is with the help of a electrical conductivity that is measured into a, we have to say decisiemens per meter. Whenever there is a uh, 0.8 decisiemens per meter is there, that is nothing but the 5% of a saline, or we have to say that the electrical conductivity of that one, isn't it? Here conductivity is measure of ability of a solution to conduct the electricity. To carry a current, solution must contain charged particles or ions. Most of the conductivity measurement are made in aqueous solution, and the ions responsible for the conductivity comes from the electrolytes dissolved in the water. Salts like sodium chloride, magnesium sulfate, acids like hydrochloric acid, acetic acid, and bases like sodium hydroxide and ammonia are the electrolytes. Conductivity is not a specific, it measures the total concentration of ions in the solution. It cannot distinguish one electrolyte or ion from another. Not all the aqueous solutions, my friends, have a conductivity. Solutions of a non-electrolyte, for example, sugar or alcohol, they cannot miss through, never electricity get, we have to say that pass. So, sugar and alcohol have no conductivity because neither sugar nor alcohol contains ions, nor do they produce ions when dissolved in a water. Conductivity measurement is an extremely widespread and useful method, especially for the quality control purposes. Observation of a drinking water quality, estimation of a total number of ions in the solution, or direct measurement of a TDS, that is total dissolved salts, in the solution is possible with the help of this conductivity meter. The high reliability, sensitivity, and relatively low cost of the conductivity instruments make it potential primary parameter of any good monitoring program. In general, the measurements of conductivity is a rapid and inexpensive way of determining the ionic strength of solutions. My friends, again most important instrument, that is of a centrifuge. We know that actually we want to separate solids from the liquid, isn't it? Solid from liquid or solutes from the liquid, then it is possible with the help of this one. A centrifuge is a device used for separating particles from the solution according to the size, shape, density, viscosity of the medium and the rotor speed by applying centrifugal force. Means जर फोर्स, ज्याला आपण स्पीड म्हणतो, स्पीड जर त्या रोटरचं कमी असेल तर इट टेक्स द टाईम, इजंट इट. स्पीड जास्त असेल तर फास्ट आपल्याला काय होतं की त्याच्यामध्ये पार्टिकल सेपरेट करता येतात सोल्युशनमधून. इन ए सोल्युशन पार्टिकल्स या हायर डेन्सिटी दॅन सॉल्वंट गेट सेडिमेंट्स. म्हणजे आपल्याला माहिती आहे जेवढे जाड मोठे सोल्युट्स असतील ते इमिडिएटली त्या टेस्ट ट्यूबच्या बॉटमला सेंट्रिफ्यूज करत असताना येतात. त्याच्यानंतर त्याच्यापेक्षा कमी साईजचे, त्याच्यानंतर किंवा सगळ्यात कमी साईजचे टॉपला येऊन सेडिमेंट्स होतात, सेटल होतात टेस्ट ट्यूबच्या साईडला. सो दॅट इज ॲक्च्युअली प्रिन्सिपल. द ग्रेटर द डिफरन्स इन द डेन्सिटी द फास्टर दे सेपरेट. समजा आपण द सोल्युट्स आहेत त्याच्यामध्ये पार्टिकल्स आहेत, पार्टिकलच्या साईजमध्ये व्हेरिएशन असेल. म्हणजे एक्झाम्पल म्हणून घेऊया आपण, एखादा पार्टिकल आहे तो ज्वारी आपली आहे तेवढा ग्रेन एवढा आहे आणि त्याच्यापेक्षा एकदमच छोटा आहे, तर त्याच्यामध्ये व्हेरिएशन आहे, दोन्हीमध्ये खूप अंतर आहे त्यांच्या साईजमध्ये, मध्ये लवकर सेटलमेंट होशील. पण ज्वारी त्याच्यानंतर थोडं बारीक, तर थोडं बारीक, एकदम थोडं बारीक, एकदम थोडं बारीक असं असेल तर इट मे टेक्स टाईम. दॅट इज नथिंग बट द ग्रेटर द डिफरन्स इन डेन्सिटी द फास्टर दे सेपरेट. If there is a no difference in the density, the particles stay steady and there is a need to use the centrifuge. अशा वेळेला आपल्याला सेंट्रिफ्यूज वापरावा लागतो. सेंट्रिफ्यूज इन्स्ट्रुमेंट इथं ठेवलेलं आहे. परीक्षेमध्ये ही सगळी इन्स्ट्रुमेंट तुम्हाला स्पॉटिंग किंवा आयडेंटिफिकेशनला ठेवली जातील. तुम्हाला त्या इन्स्ट्रुमेंटबद्दल प्रिन्सिपल लिहावं लागेल त्या इन्स्ट्रुमेंटचं आणि त्याचा यूज पण लिहावा लागेल की हाऊ दॅट वर्क्स, दॅट इज वर्किंग ऑफ इन्स्ट्रुमेंट. यू हॅव टू राईट प्रिन्सिपल, वर्किंग ऑफ इन्स्ट्रुमेंट अँड again uses of that instrument. So in three way you will have to study of this one. If centrifuge can separate molecule having even a tiny differences in densities using a centrifugal force. The force created, that is a centrifugal force, that is responsible for the separation of a particles of having according to their size, shape, density and viscosity. A centrifuge used in industries, research laboratory in separate particles or macromolecules like cells, subcellular components, proteins, nucleic acids that can be separated by using this centrifuge machine. My friends, with this I have to say thanks, but before that one, you will have to keep in mind the experiments, physiological experiments, demo experiments, whatever we have studied. Again, some instruments also we have studied. So you will have to remember those things very well, and that things will help you to understand the, first of all, why to do, how to do, where to do, and which type of precautions we have to take at the time of setting up the physiological experiments. With this, today I'm going to stop here. Thank you very much.